Hi everyone, my name is Aparna Bhaduri and I am a new faculty in the Department of Biological Chemistry and will be starting in January 2021. I'm very excited to take rotation students in the winter and spring and am excited to share with you now some of the directions of the lab. In our lab, we're interested in understanding how we make a human brain. Specifically, how does an initially uniform neural stem cell pool generate the vast number of cell types that exist in the cortex? To do this, I have previously and will continue to use single-cell omics technologies, and I seek to use organoid models as a way of genetically modifying some of the cell fate specification factors that may be important for these processes, as well as using the counterexample of glioblastoma, an adult aggressive brain cancer that reactivates many of the developmental programs that we initially see during normal cortical development. To perform this work, the lab will perform comparisons and manipulations across samples and use a variety of bioinformatic approaches, as well as explore different axes of variation, such as cell type heterogeneity, gene expression, as well as components of metabolism to better understand the regeneration capabilities of these neural stem cells, their developmental biology trajectories, as well as the cancer biology aspect of cell diversity that is reactivated in an aberrant context. For two example rotation projects, I'd like to first introduce you to some of the data sets that I have made as a postdoc, where I have been leading an initiative as part of the Brain Initiative to create an atlas of the developing brain. And as you can see here, using single-cell RNA sequencing, we've identified a number of single-cell populations that comprise the cortex. And if we look at them across the entire cortex, we see that some of these populations are area-specific. In this and a previous publication, we identified that early born neurons already express specific identities of parts of the cortex with unique specializations, such as the motor cortex or the visual cortex. And in a comparison with cortical organoids, we identified that they are capable of expressing these specific area signatures. This work was performed using gene network analysis, but what we discovered was that not only do the cortical organoid neurons also have these unique area signatures, but they are stochastically determined and expressed, making the cortical organoid the perfect model to start to understand how individual factors in the primary developing cortex actually regulate the emergence of these areas. As a rotation project, one could start using the data to prioritize candidates and then start performing knockdown experiments, either on a single candidate basis or with a screening approach to characterize whether we can direct organoid neuronal identity one direction or another. As I mentioned, the lab is also interested in understanding developmental trajectories in glioblastoma. And in previous work, we have been able to label a number of populations within the tumor bulk using single-cell RNA sequencing by developmental or adult cell types. And one of the cell types that we saw was reactivated is the outer radial glia, a cell that undergoes what's known as a jump and divide or mitotic somal translocation, as you can see in this video, which may have a role in propagating the tumor and creating tumor recurrence. A second project could start to look at this and other populations to better understand where did the glioblastoma come from, what genetic events in which cell types create the tumor. And to this end, I do have a data set that will help us answer these questions better, as well as understanding where is the tumor going. So how can we take individual cell types that exist as part of the tumor that we have already labeled and put them in the context of a cortical organoid, where we have seen that it is possible to recapitulate the tumor heterogeneity and better understand which of the cell types really give rise to the ultimate tumor populations that make those recurrent tumors and kill patients. There's a lot more to discuss on both of these projects, and I would love to have these conversations with you. So if you are interested in learning more about the lab, please visit my website at bhadurilab.dgsom.ucla.edu or contact me directly and we can have a conversation at aparnabhaduri01@gmail.com. Thank you so much for your time, and it will be wonderful to see graduate students in the Bhaduri Lab come 2021.